Okay, so we're going to have a look at some aspects of inheritance and sex and chromosomes to start with. Okay, so we need to just remind ourselves of some definitions before we go on with the content. I've got some videos that actually will help with some of this as well, because it is quite confusing. What we can see on the screen is a typical animal cell. And you can see the dark purple bit there is the nucleus. So the nucleus contains or houses the genetic information. So the genetic information is found as chromosomes, and you can see that circled there in a pink circle. That's a chromosome. Those chromosomes together make up the genome, and that's the entire DNA of an organism. So chromosomes are the structures that our DNA is ordered into. It's how they're packed up. They're structures made out of DNA, and that DNA is wrapped around proteins called histones. That basically helps to keep the DNA from getting all tangled up. There are small sections within the DNA that are known as genes, and it's these genes that code for specific proteins that give us certain characteristics. So a gene is a short section of that long genetic material, that double helix that we're familiar with seeing. A gene is a short section of that, which will code for a specific protein, which we've had a look at in previous units, how the genetic code relates to protein. So during reproduction in humans, we have eggs and sperm, and each egg and sperm contains half the normal number of chromosomes that we would expect to see in a normal cell. So this is what you'd expect to see in a normal cell. In a human, there are 46 chromosomes in total, 23 pairs, and each pair contains a chromosome from mum and a chromosome from dad. And during reproduction, you receive half of the chromosomes that you're going to get from your dad in the sperm and half that you're going to get from your mum in the egg. So the egg will always carry an X chromosome. So this last pair of chromosomes will be either XX if you're female or XY if you're male. The egg will always carry an X chromosome, and the sperm can be either X or Y in the place of this last chromosome, either X or Y. So when the sperm meets the egg, the nuclei fuse. So that means the nucleus of the egg joins together with the nucleus of the sperm. They fuse together, and those 23 chromosomes from mum plus the 23 chromosomes from dad make up a full 46 set of chromosomes in that newly formed cell, which we then call a zygote. So the egg will always have an X, and the sperm may have an X or a Y, and that determines the sex zygote then. I'm just going to show you this little video. It's quite a nice one showing the chromosome structure. Let me make sure the sound comes through. Okay, so it's quite a nice little one showing how the chromosomes are wrapped up and form this DNA structure.
What is a chromosome? In previous animations, we learned that DNA is a spiraling chain-like molecule made up of 4 different types of nucleotides. Scientists have labeled them A, C, T, and G. We also learned that a gene is a long stretch of DNA, a specific sequence of A's, C's, T's, and G's that code for something. What does a gene code for? Usually a protein or a group of proteins, though there are a few exceptions. In multicelled creatures like ourselves, proteins, which are coded for by genes, interact with other proteins and molecules to make up living cells. Cells make up tissues, make up organs, and organs ultimately make up entire living creatures. Individuals have different traits, in this case differences in beak shape, largely because of differences or mutations in their genetic code. These differences can either cause changes in a specific protein shape, and therefore its function, or mutations can change when or how much of a particular protein will be produced. Now that you understand these 3 terms, it is now time to talk about chromosomes. A chromosome is an entire chain of DNA along with a group of stabilizing proteins. You may have seen images showing DNA trapped inside a chromosome like this. If you were to unravel a chromosome, you would see that it's made of a thread, like a spool of yarn. Zoom in again and you'll see that fiber is a collection of protein orbs called histones, which are wrapped with a string-like structure. Zoom in on that string and you'll find that this is the chain of DNA. It is extremely long, millions of nucleotides long, and often contains hundreds of genes along its length. In this image, we're looking at a chromosome in its condensed, yarn-like form, but chromosomes are only packaged tightly like this when a cell is reproducing. This structure helps with durability as the cell is splitting in two. For most of a cell's life, however, chromosomes exist in a loose, noodle-like structure safe inside the nucleus or centerpiece of a cell. Here is a photograph of an entire set of human chromosomes taken from a cell right as it was getting ready to reproduce. Your genome, the entire collection of genes that help make you who you are, written across 23 pairs of chromosomes. That's 46 altogether. With very few exceptions, each cell in your body contains its own full copy of your chromosomes, your entire genetic code. Eyeball cells only use the eyeball cell genes, the rest are turned off. Liver cells only use liver cell genes, the rest are turned off, and so on for each cell type. One member of each pair of your chromosomes came from your mother, the other came from your father. If you were to examine the two individuals of a pair, you'd find that for the most part, both contain the exact same genes in the exact same locations. If you were to zoom into the actual genetic code, however, you would find slight sequence variations between them. These are the results of mutations. Most of these mutations happened long ago and have been passed down from parent to child for many generations. Others, however, are completely unique to you. These mutations occurred either in the sperm or egg, which fused to make you, or they occurred early on in your development, making it into all or many of your cells as you matured. These unique mutations represent brand new genetic information, sequences of DNA that may have never been seen before in the history of the cosmos. So, to sum things up, what exactly is a chromosome? A chromosome is an entire chain of DNA along with a group of stabilizing proteins. Humans typically have 23 pairs of chromosomes in each of their cells. That's 46 altogether. In your body, one member of each pair came from your father, the other from your mother. For most of a cell's life, chromosomes exist in a loose, noodle-like structure. Chromosomes are copied and then condensed like spools of yarn for durability when a cell prepares to reproduce by splitting in two. I'm John Perry, and that is a chromosome stated clearly.
Okay, so what we're going to have a look at now in today's session is how we get from that full cell into the egg and the sperm. So we're going to have a look at how we get to those 23 chromosomes from mum in one egg and 23 chromosomes from dad in one sperm. That's what we're having a look at today. So as we said, females have 22 chromosomes plus 2 X's, and males have 22 chromosomes plus an X and a Y, and that determines sex. But before we go any further, let's just remind ourselves that these aren't the only outcome, these aren't the only scenario. So in mammals, primary sex determination is chromosomal. It's not usually influenced by the environment, but in some cases, there can be differences from this XX and XY genotype. So in most cases, the female is XX and the male is XY. That's what we would call a typical example. So the Y chromosome is a chromosome that carries the genes which encode male phenotypes. So it encodes genes that allow testes to develop rather than the ovary instead. So you're only getting one or the other. But there are variations of this XX and XY genotype. So you should be aware of this, particularly because you're on access to medicine and healthcare courses. So you will come across it in those professions. So here's a karyotype. This is where we see chromosomes. So here's chromosome 1, one from mum, one from dad, chromosomes 2, one from mum, one from dad, chromosomes 3, one from mum, one from dad. And then instead of having XX or X and Y, this individual is XXY. So they have an additional X chromosome here. So that's an unusual phenotype. This is known as Klinefelter syndrome, and you can see an individual here with Klinefelter syndrome. They are considered male because they have a Y chromosome, but they are infertile. They have erectile dysfunction and quite a female body shape. So broad hips, almost breast development, not to the extent you'd see in an adult female, but not a typically male chest pattern, poor muscle tone that you'd normally expect to see more pronounced in a male. You can have a person with up to 5 X chromosomes and 1 Y chromosome. They'd still be considered male. It is possible to have an individual with only a single X chromosome. So instead of being XX or XY, they are just X. They develop as a female, make ovaries, but the follicles can't develop and be maintained. So release the eggs. A second chromosome is needed for that. This is known as Turner syndrome. These individuals are infertile and they have a kind of broad web-shaped, webbed neck. They have some identifiable features as well. There are other conditions where one of the autosomal genes, so that means not an X or Y, one of the other chromosomes 1, 2, 3, 4, 5, up to 22, becomes involved in sex determination. There's a gene called SOX9. Humans that are XX have an extra copy of this SOX9 gene, and then they develop as males, even though they are XX. Individuals having only one functional copy of the gene have a syndrome involving lots of different organs and skeletal structures. You can see for the abnormal skeletal development of the individual in the pictures. So about 75% of patients with this syndrome, XY patients, typically chromosomally male patients with this syndrome, will develop phenotypically as females. So they will look female despite being XY, or they might develop as hermaphrodites. So both sets of reproductive organs develop. So it appears that this gene is essential for testes formation as well. So it's not black and white. It's not XX or XY. You will see differences in sex. And it's worth explaining at this point while we're talking about chromosomes and sex determination from the chromosomes. There is also the possibility that the hormonal regulation can affect the sex phenotype that develops. So there's androgen insensitivity syndrome. So we have XY individuals, so chromosomally male. They have testes that make testosterone. However, they don't have the receptor protein. So although they're making the testosterone, they're not responding to it. They don't have the receptor to notice that it's being produced. As a result, they are able to respond to estrogen that's made in their adrenal glands. They develop a female phenotype. This individual is XY, is chromosomally male, but unable to respond to the testosterone produced by their testes, which can be internal. They just end up developing and looking like a female, and they do not know more often than not until they seek fertility treatment, quite often, these individuals. So they have a distinctly female appearance. They do not have testes, but they'll have the tissue internally or somewhere within their system quite often. They develop looking phenotypically normal women, but they're sterile because they lack a uterus and oviducts, and their testes are in the abdomen. Although we're talking about chromosomal development and talking about X and Y chromosomes, be cognizant of the fact that is not always the case. It can happen that individuals are outside of this classification. So what we're talking about today is this process called meiosis. Meiosis is a type of cell division, and we've talked about cell division before where we've had mitosis. So mitosis is a type of cell division where we're just making new cells. We do it all the time: skin cells, bone cells. We need to replace broken, damaged, worn out cells, red blood cells. So we're just making exact copies. So if we need to replace skin cells, we need an exact copy. We need another skin cell. If we're replacing blood cells, we need to replace an exact copy. So that's pretty easy. It's just a copy and paste, essentially, mitosis. But we're looking today at something called meiosis, and this is different. This is how we get the egg and the sperm. It's different because we need to go from a normal cell, which has got 46 chromosomes, one copy from mum, one copy from dad, down to an egg or a sperm cell, which has only got 23 chromosomes. So it's a bit of a fiddly process. And it can be a little bit tricky to understand because of the way the chromosomes have to make a copy of themselves before the process starts. Hopefully, we can get through it. So I'm going to give you a brief overview, but I'm going to show you some of the videos. Because it's a moving process, I think it helps to watch the videos. We start off with a normal cell, which has got 2 copies of every chromosome. I'm only showing you 4. So we've got 2 copies of every chromosome, one from mum, one from dad. What happens before they divide is they duplicate, but they stick together with the copy of themselves. Then they line up in the middle of the cell. They switch a bit of genetic information, more on that later, and then they'll pull apart. And then they pull apart again. So we end up with half. Look, we've got 4 chromosomes there, 2 here. We end up at the end of this cell division process with half of the number of chromosomes that we started with. Not only do we end up with half the number of chromosomes that we started with, that these cells here, these daughter cells we call them, are all genetically different. So if you look here, you can see we've got grey and red chromosomes. In here, we've got a mix and match of grey and red. And that's because we've had a bit of switching, shuffling of the genetic material through this process of cell division. So we're going to have a look at this. This is just a very summarized overview. We're going to have a look at some videos to try and explain this process today. So if we were producing eggs and sperm with the same number of chromosomes, the diploid number of chromosomes, as a normal cell, as the starting cell, which is 46 chromosomes, each time an egg and a sperm fuse together, we would be doubling the number of chromosomes. So we'd go to, what would it be, 92. So we'd have 46 chromosomes in the egg, 46 in the sperm. They join together, and then we'd have 92 chromosomes in the new cell, the zygote, which would then become fetus. So that's not good. We can't do that because each generation, the number would double again, because then we'd be adding 92 to 92, and that offspring would have, 184 chromosomes. So we couldn't just keep doing this. It would be an impossible situation. So we need to reduce the number of chromosomes by half for the egg and the sperm, so that when the egg and the sperm fuse together, when they're involved in fertilization, we have the correct number of chromosomes. So we call meiosis a reduction division because we're reducing the chromosome number to half. It's haploid. So when the egg and the sperm do fuse together, the nuclei fuse together, the number of chromosomes is what we expect it to be, 46 in humans, and we call this the diploid number. So the diploid number, the number of chromosomes we find in all cells, pretty much, is 46, 23 pairs. Obviously, we've seen some examples which this wasn't the case, the sex chromosomes a minute ago, but that's unusual. And this happens because they all come from a zygote that will then divide by basically mitotic divisions, which is just a copy and paste of cells. And the gametes contain, that's the egg and the sperm, contain 23 chromosomes because they're all produced as a result of this reduction division, this meiosis process that we're going to look at in a moment, which takes place in the ovaries and the testes. So I'm going to pop this little video on. It's quite a nice little explanation. Pay attention to the chromosomes, what it shows you what's happening with the chromosomes at the start, because it will help you to understand how that reduction division takes place without losing any genetic information. Okay.
In this video, we're going to take a look at how the process of meiosis works and at how it forms gametes. Now, you might remember from our previous video that sexual reproduction requires gametes, which we sometimes call sex cells, and include things like sperm cells and egg cells. The important thing about these gametes is that they only contain half the genetic material of a normal cell, so we can call them haploid cells. Then when 2 of these gametes combine, they'll end up forming a normal cell that can go on to grow into a new organism. And because that normal cell will have 2 sets of genetic information, one from each parent, we call them diploid cells. In order to make these gametes, though, a cell needs to undergo meiosis, which is a few different steps that you need to know about. First, though, let's just quickly recap chromosomes so there isn't any confusion. If we were to take a human and look inside the nucleus of any cell, we'd find these things called chromosomes, which contain genetic information. In fact, we have 23 different types of these chromosomes in almost every cell in our body. In this cell, we've only shown 4 to save space, but just imagine there'd be 23. And for each of these 23 types, there's actually 2 different copies. One set is from the father, shown here in blue, and one set is from the mother, shown here in red. We can call the ones from the mother maternal chromosomes, and the ones from the father paternal chromosomes. Because we have 23 different types of these chromosomes, and 2 copies of each of our cells is going to have a total of 46 individual chromosomes. Getting back to meiosis, the first step is to replicate all of the cell's DNA by replicating all of these chromosomes. This doesn't form entirely new chromosomes as such, it just adds an extra arm to each of the chromosomes we already had, so that they all become an X shape like this, with one arm being the original chromosome and the other arm being the copy. Next, these 2-armed chromosomes all line up along the center of the cell in their pairs. So here we have chromosome 1 from mother and father, then chromosome 2 from each parent, then 3, and so on. Importantly though, which one is on the left and which one is on the right is completely random. So if we looked at 2 more cells undergoing meiosis from the same person, the chromosomes in the first one might look like this, and the other one like this. It will always be pair 1, pair 2, pair 3, and so on, but the left to right order will be random, and so it will be different each time. And this point is actually really important, because it means that in the next stage, when the chromosome pairs are pulled apart and the whole cell splits in 2, the chromosomes are going to be randomly distributed, with each half of the split cell getting a different combination of maternal and paternal chromosomes, which means the DNA in each of those 2 cells is going to be different. Next, we have the second division, and for this, the chromosomes all line up along the center of the cell again, but this time, it's the 2 arms of each chromosome that are pulled to opposite sides of the cell. And when the cell is ready, the cells then divide in half again, leaving us now with 4 cells. Now, one of the strange things to understand here is that even though a couple of these gametes might look the same, they're actually all genetically unique, because during the process of meiosis, there are tiny changes to each of them. So what we have overall is 4 genetically unique cells that we can call gametes. And remember, there are actually 23 chromosomes in each, not just the 4 that we've shown in our example. So in male animals, these would probably go on to develop into sperm cells, while in females, they'd develop into egg cells. Then if a sperm and egg fused during fertilization, they'd form a diploid cell that could divide by mitosis over and over again until it forms an embryo, and then a fetus, and then finally a fully grown organism.
One of the really important things there was that shuffling of the chromosomes randomly, when they said half goes into one cell, half goes into the other cell. So this bit here, this random order. So you can see it goes, this one's got this half here, it's got more blue chromosomes than this one, which has got more red chromosomes. So you can see that there's not an exact division of the chromosomes from the maternal and paternal sides. So we're getting differences in these egg and sperm cells that are produced at the end. So the daughter cells, we call them. This is called independent assortment. Depends on the order of these chromosomes. It's really important. It's why you're not identical to your siblings. Yes, you might look quite similar to them because you will have some similarities, but it explains why you don't look exactly the same. And it's why you're exactly identical to either of your parents as well. Okay. So it's a source, this process is a source of variation between individuals. So what's actually happening in meiosis is we start off with our chromosomes, we've only shown 4 in most of our examples. And then what happens is the nuclear membrane breaks down. So there's no nucleus then. Everything's just loose in the cell. And the chromosomes make copies of themselves. So rather than being made up of one arm, they're made up of 2 arms. Then they will line up in the middle of the cell. And this phase is called metaphase when they line up. But they will actually, before they line up in the middle of the cell to separate, they'll just pair up. They'll pair up during prophase. As they're paired up, they will swap a little bit of genetic information over. So we get this patchwork appearance of our chromosomes where they're sharing DNA. So metaphase, they line up in the middle of the cell. And then during anaphase, they move apart. So metaphase middle, anaphase apart. And what happens then is we have different combinations. This is after the first division. We have different combinations of genetic material in these 2 cells. We've got random assortment of the chromosomes. It's like a shuffling of our cards, shuffling of the decks, if you like, of our cards, of our genetic makeup. So that's the first division. Then what will happen is new spindles form, which help to separate the chromosomes. They line up in the equator again, and the chromatids will move apart. So the X is pulled apart. And we end up with our 4 daughter cells, which are haploid, half the number of DNA compared to the original parent cell. And once the cytoplasm reforms and these cells are full, they're ready to be converted into egg and sperm. They're all genetically unique because of the independent assortment when they are shuffled into different cells, and also because of this crossing over that occurs. So which gamete is used for fertilization is also really random. So if these are going to be eggs, which egg gets released each month is random. If these were going to be sperm in the male, which sperm gets to the egg is random. So there's always quite a random chance of each individual having certain characteristics which it inherits from either parent because of this random shuffling and crossing over that we're getting in both of these steps. So let's have a look at these laws. So Mendel was a scientist who was working on inheritance. He has this law of segregation. It states that every individual has 2 alleles for each gene, and these alleles separate or segregate during gamete formation so that each gamete receives only 1 allele for each gene. You get 1 copy for each gene in the egg or the sperm because we only have 23 chromosomes as compared to our normal cells, which have got 46 chromosomes. So we've got 2 copies of each gene. This happens during meiosis when the chromosomes are pulled apart into these separate daughter cells. And as a result, each egg or sperm ends up with just the 1 copy of each gene or the 1 copy of each chromosome, 23 rather than 46. So that's one way of introducing variability. Another way is the fact that the inheritance of an allele for one trait doesn't mean you necessarily inherit another trait. This is if the genes are quite closely together, that you've got more chance of them being inherited together, but each gene that you inherit is independent, essentially. It doesn't mean just because you get, I don't know, someone's blue eyes that you're going to get their brown hair as well. Okay, so each trait is independently inherited because of all of this shuffling. This happens just because of the way that these chromosomes are pulled apart during the first separation in metaphase 1 of meiosis. It's random, the way these chromosomes line up and get pulled apart. So essentially, this is a really good thing at a population level. It doesn't really matter, particularly to individuals. You just turn out looking the way you turn out, end of story. But at a population level, this is really important because it means there is variation within the population. So that means if there's an outbreak of a disease, and which is lethal to a lot of the population, think the Black Death in Europe, Some individuals won't die from it. Some individuals might have a genotype that allows them to survive because of this genetic variability. So this is very important at a population, this level of variability, this shuffling of the genetic code. So organisms show variation within the species because of this crossing over that occurs here during this prophase 1. It ensures that each gamete is a little bit different. So instead of having all the grey or red chromosomes, you can see there's mixtures of grey and red. There's different combinations of genetic material in the daughter cells compared to the parent cell here. So it ensures there's variation. It keeps the variation within the species, allows for better opportunities, different combinations of genes, which allows survival. And it's this crossing over phase that's really important. Also, sexual reproduction, because we don't reproduce by cloning, sexual reproduction will also introduce variation because there'll be new combinations of genetic material when an egg and a sperm from different individuals combine as well. And the combination of the gametes is random because, like we were saying, we don't know which one of these eggs is going to be released or which sperm is going to get to the egg. And also these mutations that we were mentioning in the video a moment ago, these are random changes in the genetic material which are found within our genetic code, within our genome. They are also a source of variation, which may allow for different mechanisms of survival of different diseases, for example. Another source of variation that we need to be aware of is the environment. This can also affect our genes because our environment can determine how easily a gene is expressed. That's called epigenetics. For example, a diet can really affect growth and repair of damaged tissue. So if we've got a good diet, we're much likely to have all the nutrients we need to repair damaged tissue. Depending on our environment, toxins, stress, or things like that can really impact which genes are expressed as well. I'm going to leave these on here, but I'll skip through them. I'm not going to go through them now, but they are up there. You can have a look. Some of these relate to stuff that we studied in other units, but it's still relevant. But I wanted to show you this video. It's a more detailed one of meiosis, showing all of those prophase, metaphase, anaphase, crossing over phases. It's much better to show it as a video because it is a process of moving of chromosomes rather than looking at the static slides. It's a little bit of a longer video, this one. I think it's 7 minutes or something, but it's worth watching because it is quite a clear one. So I'll put this on for you now.
Have you ever wondered how two siblings can have the same mom and dad and still look so different? Today we're going to talk about a process that makes that possible, a process called meiosis, not to be confused with mitosis, which sounds unfortunately similar. Mitosis makes identical body cells, like your skin cells and stomach cells. Recall from our mitosis clip that since it makes identical body cells, mitosis is important for growth and for repair of damage, or to replace worn-out cells. But not meiosis. Meiosis is a process that contributes to genetic variety. Meiosis also doesn't make body cells. It makes sperm and egg cells, otherwise known as gametes. You might recall that humans have 46 chromosomes. That's how many chromosomes most body cells in your body have. But there are some human cells that don't have 46 chromosomes. Human sperm cells and egg cells have 23 chromosomes. Why the number difference? If a sperm cell has 23 chromosomes and an egg cell has 23 chromosomes, when they come together, that makes 46 chromosomes. That will allow a newly formed fertilized egg to develop into a human. Meiosis is what we call a reduction division because you have a starting cell that has 46 chromosomes, and your ending cell, the sperm and egg cells, have only 23 chromosomes. Before we can start getting into the stages of meiosis to make gametes, we have to remember what happens before meiosis can even start. This also happens before mitosis. It's the stage known as interphase. And if you remember interphase, it's when the cell is growing, it's replicating its DNA, it's carrying out cell processes, and just like mitosis, interphase happens before meiosis is even going to start. So the starting cell has 46 chromosomes. And you have to duplicate those chromosomes in interphase before meiosis starts. That basically means you are duplicating your DNA, since chromosomes are made of DNA and protein. Because we tend to count chromosomes by the number of centromeres present, when the 46 chromosomes duplicate, we still say there are 46 chromosomes, because the sister chromatids are still attached, and we are actually counting by centromeres. So 46 chromosomes here, they replicate in interphase, and you still have 46 chromosomes in this picture. But you went from 46 to 92 chromatids. Little tricky there. Okay, so interphase checklist done. Now we can move into meiosis. You might remember the mitosis stages PMAT. The P was for prophase, the M was for metaphase, the A for anaphase, and the T for telophase. And the good news is that in meiosis, you still use those terms. But because meiosis is actually a reduction division, you're going from 46 chromosomes to 23, which means you actually divide twice. So instead of mitosis, where you divide one time and do PMAT one time, in meiosis you're going to divide twice and therefore do PMAT twice. And because of this, in meiosis, you put numbers after the phases to indicate whether you're in the first division or the second division. So let's start with the very first step, prophase 1. This is where the chromosomes are going to condense and thicken. They are actually going to line up with their homologous pairs. The word homologous means that the chromosomes are approximately the same size and they contain the same types of genes in the same locations. It is during this prophase 1 that this amazing process occurs called crossing over. This is actually a really awesome process because this is when the chromosomes, they're lined up in homologous pairs and they have this way that they can transfer their genetic information and exchange it between each other. It's like these chromosomes flop over each other and they do a little genetic information exchange here. It makes for what we call recombinant chromosomes, which can eventually contribute to the variety that siblings can have even when they have the same parents. Now we move into metaphase 1. In metaphase 1, think of the M as standing for middle. The chromosomes are now going to be in the middle of the cell. It's a little bit different though from mitosis because these chromosomes are going to be in pairs in the middle of the cell. So it's not a single file line, they're in pairs in the middle. Now during anaphase 1, think of A for away because the chromosomes are going to be pulled away by the spindle fibers. Then we end with telophase 1, where you have two newly formed nuclei, and it becomes obvious that you're going to end meiosis 1 with two new cells. Cytokinesis follows with splitting the cytoplasm. But we're not done yet. On to meiosis 2. The very first step in meiosis 2 is prophase 2. It's not going to be nearly as eventful as it was in prophase 1, though, because they're not going to have homologous pairs of chromosomes. They also are not going to have this amazing process called crossing over. That doesn't happen again in prophase 2. You have your chromosomes, and the spindles are starting to form like they did in prophase 1, but prophase 2 is just not as eventful of having that process of crossing over. In metaphase 2, remember, think M for middle, the chromosomes are going to line up in the middle. This time, though, they are in a single file line. They are not in pairs like they were in metaphase 1. Anaphase 2, remember A for away, but this time it's the chromatids that are going to be pulled away by the spindle fibers. In telophase 2, you can see the nuclei reforming, and you can also see that the two cells have divided. There's going to be 4 cells forming. Cytokinesis will follow to completely split the cytoplasm. Now, keep in mind that meiosis in males produces sperm cells, and in females it produces egg cells. Because of independent assortment and also crossing over, you're going to have variety. For example, in a male, the 4 sperm cells that are produced each time, they are all different from each other. They're also different from the starting cell because the starting cell had 46 chromosomes, and the ending cells only had 23. So they are not identical to the original, and they are not identical to each other. This is going to lead to variety, a reason why 2 siblings with the same parents can look different from each other. One last thing to think about. Scientists are often looking into the process of meiosis because sometimes the chromosomes don't separate correctly. It's called non-disjunction when a cell can receive too many or too few chromosomes in the separation. This contributes to some genetic disorders, which is something scientists continue to study.
So we had a look at examples where we had non-disjunction of the chromosomes when we were talking about sex determination by chromosomal sex determination a moment ago. So we had examples where we had individuals with more than one X chromosome, for example. And we have seen on other units as well, where we've looked at Down syndrome, which is actually called trisomy 21. And that occurs because of non-disjunction of chromosome number 21. So instead of that chromosome separating properly, some individuals end up with 3, or trisomy 3 chromosomes, 3 copies of chromosome number 21. So that's what can happen when we don't get correct separation, when you can see those chromosomes being pulled apart in some of those stages. So we're going to have a look now at spermatogenesis and oogenesis, which basically spermatogenesis is making sperm and oogenesis is making eggs. So spermatogenesis is the process by which the male germ cells develop into the mature sperm. So this occurs in the seminiferous tubules of the testes. So obviously it's happening in the testes. And it begins in puberty and it continues throughout adult life. So it's a continuous process in the male. Men are, after puberty, constantly producing sperm. The entire process takes about 2 months in humans, so to go to a completely new set of sperm. In each primary spermatocyte, which is the cell that you start with, will end up producing 4 haploid spermatozoa. So the parent cell ending up with the 4 daughter cells, the spermatozoa at the end. So it is continuous, it's hormonally regulated, and it's producing genetically unique daughter cells. Remember that meiosis process gives us our genetically different individual sperm cells, and they're capable of fertilization when they meet an egg and fuse, nuclei fuse. That's the nucleus of the sperm and the egg, the nuclei fuse together. So here you can see, this is part of the testes here. You can see this seminiferous tubule, which is the site of this process here. If you actually look really close, you can see the as well, which are part of the cells in the process. It's actually quite a clear image, this, but if you're not used to looking at these sort of images, they're not very easy to see, to be honest, but you can see the actual sperms there, spermatids beginning to form. So there's the initial phase, the spermatogonial phase. It's a mitotic proliferation. So that means copy, paste, copy, paste, copy, paste. So we've got these diploid stem cells that are dividing by mitosis. You get exact copies when something's dividing by mitosis. Some will remain as stem cells, so they're there to be copied to maintain the germ cell population. Others will differentiate or specialize into these primary spermatocytes, so primary sperm cells. And once they've turned into a spermatocyte, you can see here, they are then committed to go down this sperm production route. So we've got this sort of stem cell group. They just make exact copies here of themselves. And then some of these become the spermatocytes. So these ones are definitely going into sperm production. And then some of these just hang around to keep making copies of themselves. So we've got this primary spermatocyte then. That's going to go through meiosis 1, where we get that first lot of genetic shuffling, that crossing over. And then we've got meiosis 2, where we end up with these haploid cells that become the sperm. So we're up to here. So these primary spermatocytes now, they're committed. They're going to go into meiosis, this meiotic phase. We're going to go into the reduction division. So we're going to go from the normal number of chromosomes, which is 46, and we're going to go to half the number of chromosomes, which is 23. We're committed in that. That's the first division, meiosis 1. That's the second division, meiosis 2. Those are our 4 daughter cells. It's a pretty rapid process to end up with these spermatids, reducing the chromosome number in half. We're introducing this massive amount of variation that we saw with the shuffling by crossing over and then random shuffling into each of these individual sperm cells as well. So each one of them ending up different to the one that you started with and to each other. Then we go into this maturation phase. There's no further division or anything, but they just need to specialize further. So these changes that happen in the specialization process include something called the formation of the acrosome. So the acrosome is an adaptation of the sperm cell. It's like the cap around this sort of head. There's a special cap which is filled with enzymes which helps the sperm dig into the egg. There's condensation of the DNA in the nucleus. So that just means it wraps up and thickens more. And this little tail flagellum will develop as well. And that flagellum is a specialist adaptation of the sperm cell, which is obviously its full motility. It's how it moves and swims to get to the egg as well, because it has to swim all the way from the vagina through the cervix up to the oviduct or fallopian tube. So it's got quite a long distance to cover as well. And it will get rid of some excess cytoplasm. There will be a lot of mitochondria in here as well. So the resulting sperm then ends up looking, we're pretty familiar with the idea of what sperm looks like. It's usually an example that they show you a differentiated or specialized cell. So you've probably seen the structure before, but it's got this head, midpiece and tail with the acrosome with the specialized enzymes in, the tail for swimming and the multiple mitochondria for energy production. Oogenesis is the process by which the female germ cells, which are sort of the parent cells, develop into the mature eggs or ova. This is obviously occurring in the ovaries. It involves the growth and differentiation. So that means specialization and maturation of these oocytes resulted in the formation of a haploid ovum. Ovum means egg. So haploid meaning half the number of genetic information, or 23 chromosomes in humans. This actually happens during fetal development. So this is happening in a female when they are in utero, in their mother's uterus. It will then pause for a long time until puberty, when this process will resume. And then one mature ovum from each primary oocyte with the remaining products forming these polar bodies is produced. The process is discontinuous and it's really closely regulated by hormonal cycles, which are quite complex in females. So it's a finite process. It's really tightly regulated. It converts these diploid germ cells, so these sort of original starting cells, which have 46 chromosomes, into these haploid eggs or ovum, which are then capable of being fertilized with a sperm. So what this will then do is ensure genetic stability, because we've got this haploid number, and also there should be sufficient cytoplasmic resources, so big cytoplasm, for early embryonic development as well, because once the egg and the sperm have fused, what happens next is that's then called a zygote. That's going to develop into a fetus, and to start with, it develops into an embryo where you make copy after copy of that original cell. So let's have a look. Multiplication phase. So we've got these germ cells. These are the primary cells, and they've got the normal number of chromosomes, 46, and they're going to undergo mitotic division during, this is the multiplication phase after we've gone through this cell division phase. This is what happens once we've gone through fertilization. So we get to that stage where we've got this cell which can be fertilized, and once it's been fertilized, we get to a multiplication phase. So egg and sperm fertilized, we've got 46 chromosomes, and they're going to copy until you've got a big ball of cells called a blastocyst. So they undergo mitotic divisions, just making copy after copy of that fertilized egg and sperm. And that blastocyst, then, that big ball of cells, will be able to develop into a fetus. Bye, everyone.